Project Catalyst presents Bench to Bedside Chats. Direct from Building 22, White Oak Campus, FDA. Brought to you by the Oncology Center of Excellence under the direction of Dr. Richard Pazner. Beyond these doors, the Oncology Center of Excellence, in collaboration with the Centers for Drug Evaluation, Biologic Evaluation, and Devices in Radiologic Health, and most importantly, dedicated people to include scientists, regulatory reviewers, and leaders, have developed multiple programs focused on improving the treatment and cure of cancer. Project Catalyst is one of those efforts intended to help the academic scientist, university life science accelerator, and oncology startup company navigate the regulatory drug development enterprise to translate your insights into drugs to treat and cure cancer. Well, good afternoon, Dr. Waring. I'm thrilled that you could take the time today to talk to Project Catalyst and discuss the S9 guidance document. And before we get started, I just wanted to give everybody a little background on what these bench to bedside chats are about. They're informal discussions where we get to kind of start with a guidance document. And today we're going to talk about the ICH S9 and FDA S9 oncology toxicology guidance. And it lets us have a little kind of a launching point to add in a little bit of additional granularity to that guidance document. We're not going to make new policy from the podium, but that we might provide some context and some more concrete ideas around what some of those sometimes statements that you can have one sentence in a guidance document and yet that implies a whole bunch. And hopefully we'll get a chance to talk about that today. I'm thrilled that we get to have you here. And I know you've been at the FDA for 10 years. And I know you probably have, I think I asked just to have a little kind of brief presentation about what the S9 guidance document is, and then we'll just go on a conversation after that. And I'll ask you some questions if we get to some places where there are some curveballs. I know some things are, that many topics are drug specific, and it's very hard to make any generalizations. And once again, everyone should realize that you need to come in for a pre-IND and an IND meeting, and that's where we can give you specific advice, but we can kind of help you interpret some of these concepts in the guidance document. So I'm going to give it to you, Emily.
Okay, it's great to be here. So ICHS-9 is the guidance that we use for non-clinical evaluation of anti-cancer pharmaceuticals. And we use it for both small molecules and biologics. There's also an ICHS-9 question and answer document. I believe that was finalized back in 2018, and that provides some additional clarification and insights to the original guidance. And so ICHS-9 was really developed to expedite getting new therapeutics to patients with cancer as quickly as possible. And so we use ICHS-9 as the starting point for all cancers. And if you're evaluating a non-advanced cancer or an adjuvant or a neoadjuvant indication, then you may need to add additional studies as appropriate based on ICHM3 or ICHS6. And if you have any questions about the indication, you can come and talk to us.
So imminently life-threatening is usually, chronic CLL, I imagine, is not probably going to fall into imminently life-threatening, but, blast crisis in CLL might. Basal cell carcinoma clearly is not going to fall into any kind of imminently life-threatening, but advanced basal cell carcinoma that's metastatic and and can't be resected. I mean, those things happen, and it probably would fall under this. So, I mean, there's some grayness in there, but you guys would try to help people understand that,
right? And we do consult with our clinical colleagues regarding each indication as to whether it would be considered more of an advanced or non-advanced indication.
And I always like to promote what my colleagues, at the FDA do. And this whole idea of trying to get drugs into the clinic sooner and evaluate them is the way that we find new paradigm changing cures. And your approach is to make sure that it's a reasonably safe starting dose place so that we can not likely have an untoward outcome, although those sometimes happen. But that is not overly burdensome to get to the first in human trial. Is that a fair assessment of what, I mean, is that how you guys like to approach it? Because that's the way I see you approaching it.
Yes. So ICHS 9 was definitely established to provide some regulatory flexibility for these indications. I can talk about, if you want, what we expect for an original first in human IND.
Absolutely.
In a first in human original IND, we generally like to see, some description of the pharmacologic activity or the mechanism of action of the drug. And in terms of toxicology studies, we expect that you would submit GLP compliant 4-week repeat dose toxicology studies in rodents and non-rodents. And of course, if your product has only one pharmacologically relevant species, then just conducting the toxicology study in that species would be appropriate.
Jump in and ask some questions here, because just to make sure that because that's pretty quick, and I think people might miss some of the nuance there. A rodent and a non-rodent species, almost always that's going to be a small molecule drug. That's one of these, we just had a presentation today at lunch, and most intelligent oncologic development is towards targeted therapies, as in we have a pathway, we have an aberration, the cancer cell is different in some fashion from a normal cell, and we're going to target that potential vulnerability. That's, whereas in the past, we had cytotoxic drugs that still had targets, they just mostly killed rapidly dividing cells in a different mechanism, but also other normal rapidly dividing cells were killed as well. But for biologics, that's where, when you talk about a relevant species, that there may be only one appropriate, most relevant species. Can you focus on that a little bit?
Sure. So, say you're developing a biologic, and just say it's an antibody that targets EGFR. So when you're doing your non-clinical studies, you want to choose a pharmacologically relevant species. So you'll want to choose a species in which the binding of your biologic to EGFR and the test species is similar to the binding of your biologic to human protein. And so we also want to make sure that that the animal species that you choose, that you can see pharmacologically relevant effects in that animal species. And that relates also to the toxicity that you'll be seeing. So we want to see relevant toxicity in the animal models. Okay.
And for the help, I know in the S9 guidance, there's essentially one sentence that talks about pharmacology. In any good drug development program out there, people are going to try to understand the mechanism of action of the drug, whether you're hitting the target, et cetera. But what is and I know I've discussed this with other the toxicology colleagues and with yourself as well. What are the expectations for pharmacology? I mean, there are some naturally derived products and or even herbal products where pharmacology might not be completely crystal clear. What's the expectation? How do you guys look at that as like, this was sufficient pharmacology?
Well, there is a pretty old guidance on the content and format of INDs. And in there, it specifies that you should provide, some description of the pharmacology and mechanism of action of your drug if it's known. So for things like botanicals, the exact mechanism of action may not be known, but in the case of those, it would be important to do, some proof of concept studies, showing why you want to give this botanical to patients with cancer.
I had this conversation with someone in the past. In the regulations, there are no clear requirements that you show that your drug has activity against tumor cells. But there is an expectation that when you're putting a drug into a patient, that there's some risk balance, the risk benefit assessment. And if your drug has substantive toxicities associated with it, and you provided no data to suggest that there's any potential for anti-cancer activity, it's an herb from the Himalayan highlands that kills mice. I mean, that drug would go on hold based on the fact that there's a risk benefit balance here that's completely inappropriate, because you provided no data to suggest that this is going to have an so we, even though it may not be crystal clear in the regulations, we anticipate and expect that we're going to see as much mechanism of action in pharmacology data and activity data in the particular tumor. Is that a fair statement?
That's fair. And I would also argue, if you haven't done those studies, then why are you pursuing the development of the drug?
To talk about the how we approach the starting dose, and there would what you guys call it, the minimal toxic standard minute, the STDDM. Go over those, because those are not always clear. Many times sponsors either pick the wrong species that they're going to base their initial starting dose on, or help people understand how to do that.
Yeah, so our standard approach for determining the starting dose is to use either 1/10 of the severely toxic dose in rodents, which we call the STD10, or 1/6 of the HNSTD in non-rodents, which we call, which is the highest non-severely toxic dose. And so for small molecules, we scale via body surface area. So that's milligrams per meter squared. And for biologics, we use just body weight or milligrams per kilogram to set the starting dose. So the highest non-severely toxic dose is the dose at which you don't see any mortality. You don't have any dose reductions. There's not like a really severe decrease in body weight.
Those species is the one I want to use, which is the most relevant for the determination of the dose, the one that's most sensitive, as in the one with the lowest dose that express those symptoms or toxicities. That would be the dose. That would be the animal.
We typically use the most sensitive species. And often we take 1/10 of the STD10. And then if that dose is not severely toxic in non-rodents, then that would be an acceptable dose to use.
Coming from a laboratory background, I always like to think I can do this. And I'm sure there are many scientists out there who think we can do this. We can save ourselves $1 to $5 million in a GLP toxicology study. We want to do it in our lab, and you're going to have to do it as a GLP study because it's in the regulations, right? So we have no flexibility in that endeavor, in that arena. And there are a lot of I don't want to tell the audience. You tell the audience you do not want to try to run a GLP toxicology study, and there's a reason that there are companies out there that specialize in this.
There are very specific, regulations and procedures. It's described in 21 CFR Part 58, and we see toxicology studies coming from academic laboratories, sometimes, but often they're not actually GLP compliant.
What do we have to tell them?
Your INDs on hold.
Can you kind of elaborate on because people ask us all the time, the MABEL approach? All of a sudden we switch our whole mindset, based on what we assume or what the suggested mechanism of action is, and we use a different approach. One that I might go, why aren't we using it for all of our, all of our dose determinations? But go ahead and opine upon.
So the MABEL approach, it stands for minimally anticipated biologic effect level, and we use this approach for immune modulating products. Especially products that have a risk of causing cytokine release syndrome or, T cell activation. And our approach to the MABEL has been evolving over time. We're currently trying to take more of a totality of evidence approach where we incorporate a MABEL, but also look at other things like PK/PD modeling. But a MABEL is usually really heavy on pharmacology data. So we often look at, EC50 values or EC20 values from in vitro pharmacology studies using human cells. We look at in vitro studies that are evaluating human T cell activation or human cytokine release. We can also incorporate in vivo xenograft studies to see where the lowest dose was that we saw anti-tumor activity in animals. And so it's more of a, it's more focused on the pharmacology and the toxicity than on the toxicology studies. But again, we are moving towards more of a totality of approach, of data approach.
When, let's say I have an antibody and it binds to a T cell, and maybe it binds to some, and presumably it probably binds to some other immune cells because cells express lots of different stuff. Is there a way that, many times the MABEL dose is small. I mean, I've had many companies go, this is 3 logs, 4 logs lower than the dose that we think we should start at. Is there a way that you would, and I tell them in the past, and I know you probably have told them, well, you do single patient dose escalations, and we try to allow rapid dose escalation in single patient cohorts to cover those lower doses, and then when we see the first grade 2 toxicity, then we go into a standard 3x3 or some other dose escalation mechanism. But is there, if I throw my drug product on PBMCs and see almost 0 or minimal compared to control cytokine release, is that sufficient to not use a MABEL approach? Or how do you guys approach that?
So if there's less risk of cytokine release, we would focus more on the totality of data while also incorporating a MABEL. One thing that sponsors have been doing recently that is a way to start at a little bit higher dose than the MABEL is if you do pharmacology studies where you look at a side-by-side comparison between your drug and a drug with clinical experience. And if you can show that they have similar activity, similar immunomodulatory properties, then you could potentially start at a higher starting dose based on the clinical experience with the other drug, if that makes sense.
Absolutely. And once again, it goes to what the statement that we made before was you guys really try to think through these, it's product specific, it's data specific, you think through, the mechanism and the concerns, and are not unwilling to look at all the data and be flexible. That's what the whole S9 guidance is about in the first place, is flexibility.
But to be clear, in order to use that approach, you would have to use both drugs together, in the same assay. You can't do cross-study comparisons.
Let's say I threw my product on some PBMCs. I saw some cytokine release. It wasn't outrageous. I'm going to use the MABEL approach. Do I still have to do the $5 million toxicology study? Is that to show for us to anticipate what the toxicities might be in the patient, so we can be pre-warned and observe for those?
If there's a pharmacologically relevant species, then we do still want to see a toxicology study. They can be informative in terms of looking at cytokine release in the animals, in terms of the timing of when it develops. But at this point, if there's a pharmacologically relevant species, then we do want to see a toxicology study.
Also expect to use it, despite what the toxicology study suggested might be the dose to start, we're also going to put in the MABEL, the concepts of the MABEL, and start at a lower dose because of our concerns.
But obviously, if you saw, a lot of toxicity in your toxicology study and it supported a lower starting dose than even the MABEL, then obviously, we would go with the lower dose.
Discuss the, and I'm going to throw curveballs here at you, different products, product types. And I know, it used to be pretty simple in the past. You got an antibody that recognizes an epitope, you got a small molecule drug, 2 species, 1 a non-rodent, and then an antibody that recognizes the epitope is the most relevant species. Presumably, there might be some nuances in there as that, it might recognize the epitope with different affinities on different species, probably want the highest affinity. And it might be important that it's expressed on similar cell types. If the epitope is expressed on dissimilar cell types in a animal versus a human, you might want to use a lower affinity, but a more representative cell expression. I'm guessing that, but you guys would use some insight into what the most relevant species is. But in today's drug development, which I think is fantastic, but these bifunctional engagers and trifunctional engagers, that's a little more complicated as to what the relevant species is when there are multiple different epitopes being recognized. And I know we don't want to be specific here at all because we're not making policy from the podium, but give a kind of a concept about how people should approach that.
These type of products definitely aren't straightforward. We would recommend that you come in for a pre-IND meeting to discuss your product before you initiate, any big GLP toxicology studies.
Not to interrupt your train of thought, but you should, if you're a small company, you are not doing yourself a favor by doing any GLP tox studies before you talk to the FDA. Don't do that. I mean, if you're a big pharma and you've done this 100 times, then you may very well know exactly what you're doing. But otherwise, have a pre-IND meeting and come in and discuss what the appropriate species selection is and dose, etc. Yeah. Sorry to interrupt. Go ahead. The multifunctional engagers and the general idea about how you would think about what the most relevant species is for, when you have 3 different epitopes that are being recognized, and they're only one of those epitopes is expressed in 3 different species. Or two epitopes are expressed in one species and one of the epitopes isn't. When I've spoken to you before, your thoughts have been to provide us with all of that data, and then your not arguments, but your ideas about what would be the most relevant, informative studies to help the FDA and the drug and the company itself, prepare for a first-in-human trial. Is that
right? Yeah. So, as the sponsor, your drug the best. So you should provide, some case as to whether you do or do not need a toxicology study for your specific product. And you should incorporate, all the relevant data, all of the binding data, any dose range binding studies that you've done, any mechanism of action and pharmacology data. And then it's important to consider also whether binding to just one epitope will induce any pharmacologic activity, or whether you need binding to all of the epitopes to see any pharmacologic activity. All of that will play into whether it's appropriate to conduct a GLP toxicology study for that product.
This is definitely a curveball here, but there are I am just thrilled with the different ideas that folks have to develop different drugs, and there are, various ways via siRNA and antisense and viral vectors, which we won't see CBER would see, but other nanoparticles to deliver some of these antisense technologies to downregulate gene expression. And then there are also aptamers, which are, essentially RNA structures that bind, act like a small molecule drug, but are really RNA. How do you guys approach, and I know there's, initiative to write a new guidance document on these, but any general thoughts that you could relay to the community about how to approach these aptamers versus antisense kind of approach?
So as you mentioned, we don't have at this time a specific guidance that's specifically for oligos or siRNAs or aptamers. We sort of view them as a hybrid between small molecules and biologics based on their size. As of now, we still follow ICHS-9 for these. It is important that you show that the species that you use for your toxicology study is pharmacologically relevant. So is there homology between your product and the target in the test species.
Antibody drug conjugates. I know the questions in the questions and answers document focuses on antibody drug conjugates. Do you want to talk about those and kind of what the expectations are for toxicology studies? What's a relevant species? What the warhead alone, the warhead on the antibody? What do folks need to do, for those kind of products?
So for antibody drug conjugates, we consider the payload to be really driving the toxicity. And so for these products, we expect that you would evaluate the whole ADC in at least one pharmacologically relevant species. And then you can provide a more limited evaluation of the payload alone. Ideally, you could include an arm in your ADC toxicology study looking at the payload alone, but there are other options as well. And the extent of characterization that's needed really depends on, the nature of the payload. Is it a novel payload? Is it a payload? Is it a microtubule inhibitor that there's a lot of experience in the clinic with a similar payload? So those are all important considerations.
That we're on the ADCs and the payload. Sometimes the payload is a alpha emitter or a beta emitter, or and those always were complicated to me. And I would look at my radiologic colleagues and ask, I'm not sure what your approach is. Can you just give an overall kind of flavor to that?
Yeah, so we do have a fairly new radiopharmaceutics guidance for oncology. So I would direct everyone to that.
Cold and hot, that's one question that comes up. Do we have to do a hot toxicology study? And the answer is kind of no and kind of yes.
It's generally, we just expect a toxicology study with the cold product.
Okay.
And then, proof of concept studies with the entire radiopharmaceutical, showing anti-tumor activity or biodistribution.
And what organs are likely to, if for some reason this had an epitope that was expressed on the brain and it all went to the brain, not a good thing, or the heart. But the question would be, and then the relevant species would be that the expression in the animal is expressed in the same places, in the same tissues.
Yeah, the biodistribution studies are important. And the starting doses for these, are quite complicated, but we do use the biodistribution studies to ensure that, at the starting dose, certain organs are only getting the amount of radiation that is tolerated. Like there are certain limits for how much radiation, certain organs can receive, like the bone marrow. And so we incorporate all of that into determining the starting dose.
Like nanoparticles. I know there's a nanoparticle guidance out there, or a couple nanoparticle guidances that were just recently released in the last couple of years. I know Stephen Oland had a whole nanoparticle drug development conference that he did through the SBIA within the past year. But that kind of goes along the idea of, many times I would be confused. What's an excipient? What's a novel excipient? What's the active? What's the drug versus the nanoparticle? If it's a nanoparticle that's targeted with an antibody or some targeting mechanism, but then there's the payload on the nanoparticle. Which part is the drug? Which is those were always, and then what needs to be what needs to be done in the toxicology studies for those various components, or is it just all the whole nanoparticle targeting agent and payload all together is the drug product, and that's what you would expect in the toxicology study? And some of that is a novel excipient because the nanoparticle is a novel excipient. Define what a novel excipient is, and because many times I think these novel excipients fall in this is not the drug, this is a excipient. But what does the FDA and what do you guys in oncology expect to see for a novel excipient?
Yeah, so an excipient is an inactive ingredient in the drug product that's not intended to have any therapeutic effect or intent. And so there is a database online where they have curated the levels of excipients in different approved products. So that's a really good resource for sponsors. A novel excipient is one that's not qualified by safety information. So we do have a guidance for excipients, and oftentimes some additional non-clinical evaluation will be needed to characterize the excipients and its safety.
Say that last part one more time, I think.
Often additional non-clinical characterization will be needed to evaluate the safety of a novel excipient. If it's being used at levels above those for which there's prior human experience, or if it's just, completely new.
Completely novel.
One option would be to do standalone studies with the excipient, or to add it as an arm into your toxicology study.
So are there can you think of any other unique product types that we see or you see where they're kind of interesting and unique, and we've had to kind of look at them differently?
We can talk about botanicals for a minute if you want. Just I mainly just wanted to mention that, the non-clinical development of them isn't necessarily always straightforward. And the extent of non-clinical evaluation that you have to do for them really depends on the previous human experience with the botanical and how your product relates to that. So, examples are if you're using it by the same route of administration or a different route of administration, are you just taking, a bottle of supplements off the shelf, or are you further processing it?
Right.
So sometimes sponsors don't realize that they have to actually do non-clinical studies with their botanical, because sometimes that's the case if it's, a new
It 1,000 times. They ran
Yeah.
it through a column and purified, particular components of that, and it's not the same herbal product. Right?
Yes.
Yeah. Okay. So we covered novel excipients, different drug classes. Is there anything that you would want to tell these, as you participate in these OCE innovator discussion meetings that we have? What would be your advice to these small companies with less experience, obviously, than big pharma, to how to approach this, how to approach toxicology?
Yeah. So, a really big thing to consider is requesting a pre-IND meeting. We always encourage you to come and talk to us. Hire a regulatory consultant, especially someone who has experience with FDA guidances and FDA regulations. If you want, we can talk about, the content of pre-IND meeting packages. So when you're submitting your pre-IND package, you don't need to submit full study reports because we won't be reviewing them in detail at that stage of development. You can provide a summary of your data, a summary of the study designs of your major tox studies if you've already conducted them. For example, how many animals did you use? Are the studies US GLP compliant?
Once again, but let's go back and focus on we're highly recommending that you never do a GLP toxicology study. Many times companies are doing toxicology studies long before they do a GLP toxicology study. It says log orders of difference in cost, right? So we would want to see that initial data, initial toxicity data that you have.
Many sponsors already do them. It just depends on how much experience you have with conducting the GLP toxicology studies.
No, exactly. For the small companies, for these academic spin-offs, we're not, our audience is not, these are, 3 investigators who decided to, take their antibody and turn it into a drug. They do not want to do, a GLP toxicology study before they discuss this with FDA. And we're not being like a roadblock. We're trying to be helpful and save them, enormous resources that might be spent, the wrong species, maybe you need any animal studies done at all, et cetera.
We don't want you to have to redo your toxicology study for sure. So it's best to come and talk to us before you initiate it, so we can, if you have any questions about the study design or appropriate species, and we can help you with that.
And along the lines of what we would anticipate in a pre-IND, Project Catalyst has a website And I will put the link up there on the video, which kind of says, are you ready for a pre-IND, and what the expectations are, what kind of data, summaries, et cetera, that you would like to see. I know that people are people, and we all have kind of different approaches. Sometimes as a reviewer, you guys like to see more data versus less, but in general, your approach of, if you submit reams and reams of raw data, more than likely based on your schedule and the resources that you have, that you are not looking through that. What's much more helpful is a condensation and an interpretation of what that data showed is going to be much more helpful to you guys.
Totally, I agree with that.
Well, Dr. Waring, I very much appreciate you taking the time to talk to us today, and hopefully some of this insight will be helpful to some of these academic accelerators and small oncology startup companies. And I look forward to seeing you sometime soon when we're all back in the office.
Same there. Have a great day.